That's my hope. Today we are going to discuss a very important topic, and that's genetics. That's genetics. Since you are most of you are from three, I will discussing in details, but not in that details. My focus it will be number one, uh, the key definitions and the key concept and how KCSE test. So how KCSE test? Number one, let's start with the introduction of genetics. Genetics is the study of heredity and variation. So that's the definition of genetics. So in exam one, I will just define the term genetics. So to the same genetics is a study of heredity and variation. What is now heredity and variation? Is variation tunasema and this heredity tunasema, what do they mean? Heredity ni passing on characteristics. We just pass characteristics. For example, saizi nikakuangalia, if kuna mahali unafanana babako na mahali unafanana mamako, kama hufanani both of them, unafanana their ancestors. Kama hufanani hata kama hata ni kidogo, meaning that you are not wewe sasa ni mtu wa umetoka kwingine, tafuta wazazi wako. That's known as heredity. You pass the characteristics. You pass the characteristics. Number two is variation. Variation is the observable differences that are seen in organism. Observable differences tu. Saizi nikikuangalia wewe uko brown, mwingine ako aje, mwingine ako tall, mwingine ako short. That's known as variation. I think you understand kwanza, first of all, the genetics, what is this? I said that genetics is a study of heredity and variation. What does the heredity means? Heredity means a passing on or transmission of characteristics from the parent to the young ones, inatoka from the parent to the young ones. So for example, saizi lazima hata saizi ukiona uzazi wako unaweza kuangalia hivi anakuambia nini, ama mtu mwingine, ai wewe unafanana baba yako ama mama yako, ama unafanana babu yako ama auntie yako. That's known as heredity. Variation ni observable characteristics. What do we observe? Ni kuona, are you tall, are you short, are you thin, uko aje? That's the observable characteristics. That's known as variation. I think you understand those terms. Another thing is the species. Species is a group of organisms that can interbreed to give a viable offspring. Species is the key word. The key word is that the viable first offspring. Vitu zingine zinaweza mate and zinaweza zaa, lakini not a viable offspring. So even when we are marking exam, the keyword that tunaangalia what is the species is, tunaangalia the viable offspring. That's the viable offspring. Offspring sasa show slight differences from their parents. So zina show slight differences peke yake, kama shape of nose, skin color, height, because of the heredity factors. So how KCSE test? How do they test KCSE? Ile kitu tumesoma. First of all, you define the heredity, variation, genetics, and the species. Explain why offspring differs from their parent. Define how offspring differs from their parent. So species refer to a group of organisms. Hiyo tulisema offspring shows slight differences kwa sababu gani? Due to the heredity factor. In case ukulizwa why offspring show slight differences, it's due to heredity factor or the gene environment. Common wrong answers. Definition of species without mentioning freely and naturally or viable offspring. Hiyo nimekuambia already. That's the common mistakes. So let's go to variation. What does this variation means? What's variation? So let's do any kwanza variation to define meaning of the variation. Already we define it, lakini tunaingilia in deep. So this variation inamaanisha differences of organisms among the same species. Just differences. Tallness hiyo. So there's a two type of variation. There's a two type of variation. There's a discontinuous variation and continuous variation. There's a discontinuous variation and continuous variation. What does the discontinuous variation and continuous variation means? And that's can you mute yourself? So discontinuous variation and continuous variation, that is a more common question in genetics. Wanakuambia differentiate between discontinuous and continuous. So what does the discontinuous variation means? Discontinuous variation is a type of variation where there is a distinct or different differences in a given characteristic without intermediate. So it's controlled by genes and the environment has no effect. Also, one thing that you have to know is in variation, there's a two things that affect. In variation, there's a two things that affect. That is the genes and the environment. That's the genes and environment. So kwa discontinuous variation, only genes affect, but environment haina effect. Kwa sababu kama gani? Kama rolling the tank. Kama rolling tank. Ability to roll the tank. Or sex, male and female. Hata uko, kama uko environment gani, kama wewe ni mwanaume, utakuwa tu mwanaume forever. Kama wewe utakuwa ni mwanamke utakuwa forever. So there's no intermediate or intermediate characteristic or without characteristic. Hakuna katikati. You are either male or female, or you either A blood group A, B, or blood group or blood group O. So known as A, B, or blood group. Or hair in the nose, or ear in the penile, or hair in the penile. So fingerprint saizi ukifunga simu yako kwa fingerprint, si kila mtu anafungua, ni peke yako tu ndo utafungua. That's known as discontinuous variation. For example, in plant is wing length, size of abdomen, an eye color or Drosophila. Ni hizo hasa. Tuende kwa continuous variation. So continuous variation is a type of variation which shows wide range of differences at the same characteristics. So hii characteristic moja na hii characteristic, unajua hii sasa ina characteristic, ni characteristic tofauti, lakini ni characteristic moja. Na hii characteristic moja there is a lot of intermediates. Hizi zingine iko na intermediate, lakini iko na intermediate. It has intermediate. What do I mean that? For example, look at height. Look at weight. For example, saizi naweza pata aje? Naweza pata mtoto ako 10 kg, mwingine ako 20 kg, mwingine ako 30 kg. Lakini all in all hiyo nini? Hiyo inaita weight. Kuna mwingine ako 1.2 feet, mwingine ako 5 feet, mwingine 3 feet, mwingine 4 feet, mwingine 3 feet, and all of them zinaitwa nini? Zinaitwa height. Skin color in human being, kuna mwingine brown, mwingine black, and all of them tunazaita nini? Tunaita color. So hii question ya continuous variation, there's a graph that's known as a normal distribution curve. So hii normal distribution curve inakaaje? Inakaa hivi. Normal distribution curve look like this. This is a normal distribution curve. For example, saizi in a class of student, nikichukua weight ya kila mtu ama height ya kila mtu mwanafunzi ambao class of 40 student, tuseme 40 student, naweza pata watoto nikiplot a graph against the number of student against their height, nitapata curve kama hii, na hii curve inaitwa nini sasa? Inaitwa normal distribution curve. This is known as normal distribution curve. So also sometimes they may ask KCSE question, plot a graph of student against height and identify the type of curve. The type of curve is known as normal distribution curve. So how do they test in KCSE? Distinguish between continuous and non-continuous, and give them example of each. So before I go, let me define another thing. Causes of variation. Nini naleta variation? What actually bring variation? Number one is the gamete formation. During gamete formation, meiosis, that's a meiosis. The following mitotic process take place. So during gamete formation, there are two processes that take places. Number one is known as group crossing over, and number two is known as independent assortment. Let me give you the example of crossing over. Sometimes during the meiosis one, is that these kuna hii kitu tulipita. So these are chromosomes of the one parent and the chromosomes of one parent. So they cross it, and the moment they cross it, they interchange the characteristics, and then sasa mtoto anapatikana ambaye kuna characteristic ya huyu na kuna characteristic ya huyu. So nimekwambia nini? Inapatikana mtoto ambaye wanazaa mtoto ambaye akona some part of this characteristic and some part of the other parent characteristic. That's known as crossing over. Number two is the independent assortment. Independent assortment is just the arrangement of the homologous chromosomes at the equator during the metaphase 1. So independent assortment inakuanga hivi. Na nimekuambia jana wakati tulikuwa tunapita hii topic inaitwa reproduction. So they arrange themselves at the equator. They arrange themselves at the equator. So these equators are followed by segregation. Segregation is separation into two different data. So hii after this metaphase one, this inakatika hizi inakatika, hii inakatika, hii inakatika, hii inakatika. So ona hii sasa inakatika into two different way. Inakuwa hivi, nyingine inakuwa hivi, nyingine inakuwa hivi, nyingine inakuwa hivi. So zinakatika hizi katikati. So these results are the daughter cells or gamete contain different genetic combination. So hii iko na genetics ya kando, hii iko na genetics ya kando. And that's how inapatikana nini? Gamete formation and lead variation. Ya pili is fertilization. Fertilization ni when the parent mate, kila moja iko na two quality. For example, in a case of human being where ovum and the sperm they mate, when they mate, the ovum contain the mother characteristics and the sperm contain the man characteristic or the father characteristic. When they combine it, they give differently. They give different qualities to be combined differently in the offspring, and that's how it's contained. Ya tatu ni mutation. What does mutation means? Mutation is just the change in genetic makeup of an organism. Just mutation, right? And it's spontaneous change in genetic makeup. So just the changes that occur in the genetic makeup. So these changes are inherited to the offspring, are inherited. So importance of variation, causes and importance of variation, it causes by gene formation to mesema, causing over and independent assortment. So in a later fertilization, I think is the mutation. Importance of variation in kama gani? Number one, individuals with advantageous traits are favored or selected by nature and strive to pass through trait offspring. So ani hii kitu tumesema tu jana. Advantageous trait, for example, kama mzazi ako na shida, mmoja ako na shida, there's a possibility that na tutaona there's a possibility that their offspring anaweza kuwa hatakuwa na hiyo shida. And that is given by variation. Number two is it ensures that propagation of the favorable or advantageous trait to the future generation, ensuring quality of population or species is maintained. So ile characteristic ambayo imepasiwa, then it is passed and it is passed to the future generation, inaendelea mpaka ifike kwa generation. So that is important, importance of the variation. Tuende kwa chromosomes, chromosomes inakaanga hivi sasa, look like this. Chromosomes look like this. They look like this. Hizi inaitwanga chromatids, tumepita jana usiku. And this is known as centromere. Sasa kama sasa imekuwa mbili, hizo zinaitwa sasa nini? Zinaitwa homologous chromosomes. They are known as homologous chromosomes. So they are thread-like structure found in the nucleus of a plant and the cell. So they are found in the nucleus of the plant and animals. Composition, they contain material known as deoxyribonucleic acid. That's in the DNA. And this DNA hasa ndo hasa watu wanaita nini? Wanaita deoxyribonucleic acid. So that's the thing of the DNA. At least leo umejua nini DNA inamaanisha nini. And I think is the structure. The structure, they are made out of the parallel strand. Na hiyo nimekuonyesha. Homologous chromosomes in somatic, they are the pair. Na hiyo nimekuonyesha, diagram nimekuonyesha. How the KCSE is tested. Label the part of the chromosomes and define homologous chromosomes. Hiyo pia ni you just name it. So the genes and DNA ndio muhimu. So these genes and DNA inasaidia nini? Kwanza genes ni nini? Genes inaitwa nini? Genes is a structure ambayo inapatikana kwa DNA. Or chromosomes, lakini ina control nini? Heredity. Na heredity tulisema nini? Heredity ni characteristics. Is just passing of the characteristic from the parent to the offspring. So genes sasa ni just a structure ambayo iko kwa DNA, inapatikana kwa DNA. So ni kitu ambayo inapatikana kwa DNA. Na hiyo kitu kazi yake ni nini? Kazi yake ni moja. It's just to control the heredity or carry the genetic information. So kuna kitu ambayo inaitwa gene loci or gene loci. Gene loci ni specific position in the DNA where the genes are found. Nimekuambia kwanza, first of all, genes ni kitu inapatikana kwa DNA. The specific position where the genes inapatikana is known as gene loci. It's known as gene loci. It's known as gene loci. I think you're umeelewa. Then the next, that is the alleles. The alleles. Alleles ni hii alleles nini? What's this alleles? Alleles is the alternative form of the same gene that control the development of a pair of contrasting trait. Contrasting trait. Meaning that, angalia this, what do allele do? Allele kwanza ni the alternative form. They are the same gene. They are the same gene. This same gene control the development of a pair of contrasting trait. Trait inamaanisha ni characteristic. So ina form nini? A characteristic that they are opposite to each other. Characteristic that is opposite to each other. So that's known as alleles. That's known as alleles. Deoxyribonucleic acid, that's a DNA. DNA is nini? DNA is just a nucleic acid or molecule or protein found in the chromosome containing approximately 30,000 genes that occur in pair. Properties. So properties of DNA, very common question. Properties of DNA, very common question. Properties of DNA, very common question. It can replicate. Tutafika kwa replication. It can inaweza inheritiwa. Also it can undergo nini? Mutation. It can undergo mutation. Role of the DNA ni gani? DNA kazi yake nini? DNA kazi yake ni to store the genetic information in coded form. Is to store the genetic information in coded form. That's number one. Number two, it enables transfer of information and change information ambayo haijachangewa, yani kama characteristic ambayo ina changes yote to daughter cells through replication. Hiyo replication tutafika. Translate formation into characteristic through protein side. So they translate information into characteristic. The information that they have, wanabadilisha into characteristic through something that's known as protein size, and they synthesize protein. So structure of DNA, structure of the DNA, structure of the DNA. So DNA look like this. This is the DNA. So this is the structure of DNA. DNA look like this. They are double strand. So they are made up of the two strands that that are twisted around each other to appear like coiled ladder. So they appear like coiled ladder. Ladders unajua. The shape are called double helix. This shape is known as double helix. Each strand is gonna contain any nucleotide. Each strand of them, these are double strand. Now each strand is containing something that's known as nucleotide. So these nucleotide are joined together to form long strand. So nucleotide inakaanga hivi. Structure yake inakaanga hivi. This is known as nucleotide. This is nucleotide. So this nucleotide ina contain kitu tatu. The nucleotide contain kitu tatu. Three things. That is pentose sugar. That is 5 carbon. Angalia. This is a pentagon. 1, 2, 3, 4, 5. 5 sided figure is known as pentagon. So it contains pentose sugar, meaning that it has 5 carbon sugar. Number 2, it contains something that's known as phosphate molecule. Phosphate molecule. Number 3 is known as it contains nitrogenous base. It contains nitrogenous base. It contains, so kuambia structure of the nucleotide. Nucleotide consists of 3 parts. Number 1 is the pentose sugar. Number 2 is phosphate molecule. Number 3 is nitrogenous base. That's number 3. So that's nitrogenous base. So the sugar ambayo ina contain DNA. So there's a two things. There's something that's known as DNA and there's something that's known as RNA. DNA and RNA. DNA and RNA. DNA and RNA. So the DNA, the sugar in DNA, ile sugar ambayo ina contain DNA is known as deoxyribose. It's known as deoxyribose. And the sugar in RNA is known as ribose. That's the first one thing that you have to know. The sugar in DNA is known as deoxyribose. The sugar in RNA is known as ribose. So since tulisema it contains the nucleotide contains 3 things. That is the phosphate molecule. Number 2 is pentose molecule, and number 3 is nitrogenous base. This nitrogenous base it contains 4 things. Kwa DNA ina contain thymine, adenine, cytosine and guanine. Cytosine and guanine. Lakini kwa RNA base ya thymine imebadilika na uracil. Imebadilika na uracil. And that's the only difference. That's the only difference. So ina contain uracil, adenine, cytosine and guanine. Na hii ya DNA ina contain thymine, adenine, cytosine and guanine. Hiyo nitakwambia vile. So in RNA, thymine is replaced with uracil. Thymine is replaced with uracil. So acha turudi kwa note yetu. Hiyo nitakwambia. So that's the structure. Kwa hivyo pentose sugar diagram nimekuonyesha. Nitrogenous base. So this nitrogenous base sasa they pair each other. Meaning that adenine and cytosine they pair. Guanine and thymine they pair. So then uracil replaces with thymine. Meaning that look at this. DNA acha hii nikuonyeshe. Then zoom. Yes. Look at this. Pairing in DNA. Adenine na react with in a pair with thymine. Now unamwambia ujue uandike the how do they pair. So adenine pair with thymine. So adenine pair with thymine. Cytosine pair with guanine. And that's 2-2. Si ndivyo? So RNA tofauti yake ni moja tu. Tofauti yake ni moja. And that tofauti yake ni moja ni gani? Mahali ya thymine imereplacewa na uracil. Mahali ya thymine imereplacewa na uracil. That's the difference. That's the difference. So that's how this is how RNA look like. This is how RNA look like. This is how RNA look like. This is how RNA pair. Acha ni zoom hata hii acha ni zoom. Yeah. So itakuwa sa nini? Pair in RNA itakuwa sa adenine ina react with uracil and the cytosine with guanine. So that's the difference. Sometimes hii question atestiwa aje. Number one, this question is tested in 2 ways in exam situation. Question is tested in 2 ways in exam situation. Number one, unaweza kuambia nini? State the bearing of the DNA. Ukupatie DNA na wakuambie nini? Wakuambie the how do they pair it? How do they pair it? That's number one. Number two is wanakupatia pairing ya DNA for example. DNA. Ama wanakupatia nini? Pairing ya RNA. Wanakupatia nini? Pairing ya DNA ama pairing ya RNA. Na ukwambie andika sasa for example ukupatia pairing ya DNA. Ukupatia pairing ya DNA, unaweza kuambia nini? Write the pairing in RNA. 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 So kuambia write the pairing in RNA. Na wanakuchoreanga box by the way. Wanakuchoreanga box. So let me know kama unaweza chora box. Let me know kama unaweza chora box ya unaweza kuchoreanga box. Yeah. Wanakuchoreanga box kama hii. Box kama hii. So unakuwekea sasa hapo juu unakuwekea for example A. Unakuwekea A pairing with adenine pairing with thymine. Pairing thymine. Then cytosine, which is the C, pairing with guanine. Then unaendelea tu adenine pairing with thymine, cytosine pairing with guanine, adenine pairing with thymine, and the cytosine pairing with guanine. So wamekupatia hivi, wanakuchorea hivi. Then wanakuambia sasa nini? Write the bearing in RNA. Write the bearing in RNA. So kuambia bearing in RNA, the only thing that will change itakuwa sasa nini? Itakuwa the T. The T will change na uracil. So itakuwaje? Itakuwa adenine bearing with uracil. That's the RNA. Adenine bearing with uracil. Not like that. Cytosine bearing with guanine. Adenine bearing with uracil. Cytosine bearing with that's the cytosine bearing with guanine. Then adenine bearing with uracil. Cytosine bearing with guanine. So unaandika hivi. So this is for This is for RNA. This is for DNA. I mean DNA. This is for DNA, and this for RNA. This is for RNA. This is for RNA. 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 So that is how exam wanakuchorea. Wanakuchorea tu hivi. Another thing is the difference between DNA and RNA. Difference yao ni gani? DNA and RNA. DNA nimekuambia ina contain the observable sugar na hii ingine ina contain nini? Ribose sugar. Hii ingine tumesema it has diamine, hii ingine iko na uracil. Hii iko double stranded, single stranded. Hii ina found in DNA, ina found in nucleus only. Ina found nucleus peke yake. Lakini kwa RNA ina found in nucleus and cytoplasm. DNA inakuwa stable na hii ingine inakuanga unstable. Inakuanga unstable. So tuende kwa DNA process. This DNA process ambayo nilikuwa nakwambia nini? There are three things. Tunasema there is a DNA replication, transcription and translation. Replication, transcription and translation and translation. Excuse and translation. So DNA replication inamaanisha nini? DNA replication is the process through which DNA strand forms copies by unzipping hydrogen bond. So inaita replication, duplication. They form copies of themselves. Haye, kwanza uelewe, kwanza nitakuonyesha diagram. So DNA replication inamaanisha nini? They form copies of themselves. DNA transcription is the transfer of genetic information from the DNA in the nucleus, from the DNA in the nucleus to ribosome through a messenger RNA. EM inamaanisha inamaanisha nini? Messenger. Inamaanisha messenger RNA. DNA translation is the process where this mRNA they attach to the ribosome to direct the synthesis of the specific protein. Meaning that, acha nikueleweshe vizuri. DNA translation is the process where the mRNA imeformiwa, they attach themselves. Yani they attach themselves. So the moment they attach themselves on the ribosome, to the ribosome kazi yake ilikuwa nini? What is the function of the ribosome? Is to synthesize the proteins, hivyo. So this mRNA the moment imejishikisha na ama imejiattach na ribosome, they direct them to synthesize ama itengeneze a specific protein. So this thing inamaanisha nini? Kwanza sasa tumejua DNA replication, transcription and translation. Acha nikuonyeshe. So DNA, that's DNA. So acha nikuonyeshe DNA replication. This is the DNA replication. Meaning that, angalia, look at this. They form copies of themselves. So copies of themselves. Acha ni zoom. Acha nikuonyeshe copies of themselves. Zoom. So look at this. Hii kuna thymine and adenine. Guanine and cytosine. So meaning that this is the complementary new strand. So this was the parent strand. Ilikuwa tu moja hivi. Then after that they form copies. Copies I think unajua. Inakuwa mingi, yani ina form nini? Ina form copy. Number 2 was the DNA transcription. DNA transcription tunasema aje? It's just the genetic information, transfer of genetic information. Ile information tuko nayo from the genetics. Tuko nayo from the information. So DNA transcription is the process, the transfer of genetic information from the DNA in the nucleus to the ribosomes in the cytoplasm through a messenger known as messenger RNA. So they just transfer. Kazi yao nini? So these transcriptions are kazi yake nini? Just to transfer the genetic information. And transfer nini? The genetic information. Genetic information. Na hii genetic information ndo sasa wanapeleka wapi? Inapeleka eh, genetic information from the DNA in the nucleus to the ribosome. Wanapeleka to the ribosome. Wanapeleka to the ribosome through a cytoplasm known as messenger RNA. And this messenger RNA ndo sasa attach themselves. So question, kurudia question, what is the basic unit of DNA? Basic unit of DNA is known as nucleotides. Tumesoma hiyo nucleotide. Name three chemical component that's component of the basic unit. Nucleotide ina contain kitu tatu. That is deoxyribonucleic sugar, that is a 5 carbon, phosphate and organic nitrogenous base. And organic nitrogenous base. Ina contain tu hiyo tatu. Hiyo tatu. So tuende the next. So genetic terms. There is a terms that you have to know for genetics. And kama hujui hii terms itakupatia utapitia shida mingi sana answering questions and also understanding how this thing the diagram of the genes tutasoma kwa Mendelius principle. Lakini hizi size ile kitu unafaa ujue ni these terms. Number one is genotype. What is the genotype? Genotype is the genetic constitution of an organism. This organism iko na genetics kama gani? Is it tall? Is it short? Or is it in middle? Unajua either huwezi kuwa kitu ingine, hakuna kitu ingine. Either miti iko tall ama mtu ako tall, ako short ama ako katikati. So kama ako short tunatumia hii small t. Kama ako tall tunatumia nini? Hii t mbili. Kama ako katikati tunatumia nini? T mbili. Number two is the phenotype. Phenotype is the physical appearance. Yani ni kitu unaweza onekana. Saizi ni kuona. That is known as phenotype. Homozygosity or homozygous is a condition where an individual has identical or similar alleles. I think tumesoma alleles. So condition where individual has identical inamaanisha nini? Zinafanana. So angalia either ako TT ama T ndogo zote. So that's known as homozygosity. So for example, say this example. Hii inaitwa homozygosity. Also hii ingine ni homozygosity. Lakini hii iko different, angalia iko na T kubwa T ndogo. So they are heterozygous. So they have different alleles. So heterozygosity is a condition where individuals have different alleles. Another thing is the dominance. Dominance is a condition where one allele suppresses and expresses each other. That's known as dominance. Hiyo tutafika pia. Lakini just know that the meaning of the dominance is recessiveness is a condition where allele does not express themselves because of dominance. Hii dominance inaleta nini? Leta recessiveness. Diploidy is a cell has two chromosomes. Haploidy has one set of the chromosomes. Diploidy has more than two. Hybrid vigor, hii niliwaambia jana. Hybrid vigor nimekuambia is the increased performance of our offspring than the parent due to the crossing of the two superior parent. That's why inasemekana mtu akioa saizi wewe, that's why unaambiwa unafaa uangalie that person that una share my life with, si ati una fall in love blindly. That's abnormality. So unaangalia mtu kama ako na akili. So kama wewe uko na akili na bibi yako ama bwana yako ako na akili, obvious mtazamo mtoto ako na akili kuliko yeye. That's known as hybrid vigor, because of the hybrid vigor. So inshallah the next lesson tunaanza nini? Tunaanza Mendelian principle of genetics. Is there any question up to that?